Hey guys, welcome back to Pre-AP Biology at Hewitt Trussville High School, and here we are with continuing on with protein synthesis and Lecture 3, where we are going to focus in on that pretty little picture there, the ribosome, and the process of translation or making of the primary structure of a polypeptide. Alrighty, here we go. Okay, so let's start off by looking at the big picture of translation. So, once again, if this is going to be happening in a eukaryotic cell like you and like yourself, or a blue whale, or a plant, or a bug, or your cat, or your dog, this process is going to be occurring in the cytoplasm where the ribosomes are. The first step, remember, transcription, took place in the nucleus in all eukaryotic organisms. If it's a prokaryote, the process is going to be the same, except for transcription and translation can take place at the exact same time because there is no nucleus in a prokaryotic organism. So as RNA polymerase is making the messenger RNA in a prokaryote, a ribosome could attach to the first piece of that messenger RNA coming out, and it could already start decoding the messenger RNA molecule while the messenger RNA molecule is actually being built. So that's one of the main differences between prokaryotes and eukaryotes. Transcription and translation take place in different areas of the cell in eukaryotes and at different times, whereas in prokaryotes they can occur in the same place at the same time. So, the big process. Messenger RNA comes out of the nucleus. It attaches to a ribosome. The ribosome is going to read the messenger RNA molecule 3 nucleotides or 1 codon at a time, and it's going to build the polypeptide. All that means it's a sequence of amino acids, which is the primary structure of a protein. Polypeptides are the beginnings of a protein. Once it becomes properly folded, it is a protein. So, messenger RNA, once again, is being read by the ribosome, which is also, remember, the ribosome is also composed of proteins and ribosomal RNA. So it's also somewhat of an RNA-based molecule. So what's bringing the amino acids to the ribosomes? Yet another type of RNA. tRNA or transfer RNA. And as you can see, the transfer RNA does not have a codon, it has an anticodon, which is going to base pair with the codon on the messenger RNA. And each amino acid has its own specialized transfer RNA. Okay, well, not quite the next slide. Remember, what is the ribosome going to do? It's going to read the messenger RNA molecule 1 codon at a time. The codon, remember, is a sequence of 3 nucleotides found on the messenger RNA molecule. Each one of those codons codes for a specific amino acid. But do note that many amino acids have more than one codon. That is what we call redundancy, and that's going to make a lot of sense when we get to our next lecture and why that process exists. Remember, the codon is found on the messenger RNA, and the anticodon is found on the transfer RNA, and the codon and the anticodon match up. So, and again, using base pairing rules. So if I had a codon that was UUU, which stands for phenylalanine, that's the very first codon on the upper left-hand picture of the diagram, its anticodon would be AAA. Okay? Just wanted to let you know, and that would be found on the transfer RNA, and that transfer RNA would be bringing in a phenylalanine amino acid. So once again, here's a transfer RNA molecule. It is a single-stranded RNA molecule, and it base pairs with itself, and it forms a particular structure.
And as you can see, the anticodon on the bottom there. And on the 3' end, that is the amino acid attachment site. Why? Because once again, the 3' end has an open spot where a bond can form. Okay, and it's the same reason why we have to add new nucleotides only to the 3' end, because there's an open spot for it to form, to form a bond, excuse me. Okay, so there's the anticodon, and the anticodon will match up with the codon. So in this particular one, the anticodon is AAG. What would be the codon that's going to AAG? Here's other diagrams. These are a little bit more realistic. Okay, so there's the three-dimensional structure on the left-hand side, and there's our little cartoon that we like to use a lot in the Campbell's biology books, and you'll see this if you take AP biology. Okay, where we see the amino acid attachment site, and that's our little symbol there. There's the anticodon on the bottom. And then the codon on top. So, 64 of these codons are different anticodons. Okay. Well, once the transfer RNA delivers its amino acid to the ribosome, it exits the ribosome, and then it's going to go back into the cytoplasm and get another amino acid. But it does this using special enzymes. Aminoacyl transfer RNA syntase is the name of the enzyme. You don't need to know that. But as you can see, there is several docking sites on this particular enzyme. What are those docking sites called again? The blank site. What is it? Okay. And as you can see, you're going to have both ATP and the amino acid and the transfer RNA are all going to bind into this enzyme. This is a building process. We're forming a bond that is anabolic. So that, remember, anabolic processes are endergonic, which means they require energy. Where is the energy coming from? It's coming from ATP. Okay? So just wanted to show you how the transfer RNAs are replenished once they have delivered their amino acid to the ribosome. Here's our ribosome structure. They consist of two parts. So you can see there's the large subunit up on top, and then there's a small subunit on the bottom. The large subunit, as you can see, has three different binding sites in there. EPA. Okay, the A site, I like to call it the arrival site. That's where new transfer RNAs with their amino acid are going to arrive. The P site, that's our production site. That's where the proteins are going to, the amino acid from the transfer RNA is going to be attached to the growing protein chain. Okay, so P for production, A for arrival, P for production, and then E, that's going to be our exit site. Okay, easy way to remember this is EPA, Environmental Protection Agency. That's how I always remembered it. Okay, and then A is for arrival, P is for production, and E is for exit. Okay, so once again, the ribosome is going to move down the messenger RNA molecule one codon at a time, and it's going to allow for the transfer RNAs to come in and bind with their anticodon to the codon, and then transfer their amino acid to the growing polypeptide chain. So once again, just like we saw with transcription, with translation, we're going to have three steps. We have initiation. building the factory. As you can see, the messenger RNA is going to start off by binding to the small ribosomal subunit. There's going to be an initiator transfer RNA, and that's our start codon, remember? So we have the start codon as AUG, which codes for the amino acid methionine, and the anti-codon on the transfer RNA that's going to bind to the start codon is UAC. Again, base pairing rules. Okay? And then once that happens, then the large subunit will come in and bind to that with the transfer RNA in the P site. And now we have a translation initiation complex. We have a functional ribosome is what we have. And we can now start to build our proteins. And the process of the ribosome moving along the messenger RNA molecule is called translocation. So let's start up at number 1. Okay? And as you can see, we have at the very top at 12 o'clock of this diagram, we have a short polypeptide chain. We have the transfer RNA is now in the P site, and the A site is open. So the A site codon is going to allow for the proper anti-codon of the proper amino acid or transfer RNA to come in and bind with it. Once it does that, okay, then we're gonna go to number 2, and we're gonna form a peptide bond. That's just a covalent bond between 2 amino acids, okay, and the the ribosome moves down 1 codon. Okay, so now you can see the P site, the production site, is now empty. There's no polypeptide chain on there. And on the 6 o'clock on this diagram, and then the ribosome translocates down 1 more codon. The one that's in the P site goes to the E site, and it exits out, and we are back over at the start again, and with the A site being open, and for the next transfer RNA to come on in. This happens at a very, very rapid pace. We'll look at some videos in class on this, at how fast this does happen. Okay. Finally, there's termination, and once again, we get to the stop codon, UAG, UAA, or UGA, go dogs, okay, and there's a protein will come in called a release factor, and that's basically going to cause the ribosome to break apart, and it's no longer going to be a functional ribosome until another messenger RNA comes in and binds to the small subunit, and so on and so on, and the polypeptide will be released. And 2 outcomes for that polypeptide. We'll talk about that at the end. Alright, polyribosomes. Okay, so like I said, the messenger RNA is a cheap dime store copy. Okay, it's recyclable. However, it can make multiple proteins, and it can do that because multiple ribosomes can bind to a single messenger RNA molecule. And the proteins will continue to be made as long as that messenger RNA molecule is intact inside of the cytoplasm. Now, once it is broken down, then we'll stop making the proteins, and so on and so on. And different types of enzymes will break down the messenger RNA molecule and recycle all the nucleotides to be used once again. Okay, but that's a polyribosome when you have multiple ribosomes making proteins, and in this way, a cell can produce a lot of protein in a very short period of time. So there's our big picture. We have transcription occurring in the nucleus, and again, this would be in a eukaryotic organism. We have RNA processing in order to make a mature RNA molecule. We're going to take out the introns. We're going to put a poly-A tail on and a 5' cap. That's going to go out into the cytoplasm, where it's going to attach to a small ribosomal subunit. And then we're going to make a functional ribosome. We're going to translate that messenger RNA molecule into an active protein. And that's the big picture. All right, so like I said earlier, there are two outcomes of those proteins in a eukaryotic cell. If that protein is meant to be used within the cell, it will enter a special protein called chaperonin. What does a chaperone do? It kind of guides you on and protects you and things of that nature. Well, a chaperonin is a type of protein that basically guides a polypeptide to fold into the proper shape. Okay, so if that protein is going to stay inside the cell, it will generally enter one of these chaperonin proteins and be folded into its proper shape. It has a little microenvironment in there. Okay, however, if that polypeptide is bound to be exported out of the cell, then it will, instead of being translated on a free-floating ribosome in the cytoplasm, it will hook up with a ribosome in the cytoplasm, and that ribosome will then be targeted to end up on the rough endoplasmic reticulum, and there it will produce the protein inside of the endoplasmic reticulum. And once that protein is inside that endoplasmic reticulum, it can be further modified, and then eventually it will be put into a transport vesicle where it will exit the cell after passing through the Golgi apparatus.